So I want to say that this is the view of myself and some other experts from some European agencies and EMA, but it does not reflect the official view of EMA or its committees or working parties. So I'm going to talk about what the regulators' expectations are, focusing specifically on the guideline for reporting population PK analysis, and give you some examples from recent applications. And as Terry said earlier today, modeling and simulation is encouraged in several guidelines, but we don't have any guideline which states on how to do modeling and simulation. And in the guideline on reporting results on population PK analysis, there is a description of the regulatory expectations when it comes to analysis plan and report, and I will therefore focus on that guideline and give examples related to that. So on this slide, there are some quotes from the population PK guideline, which gives you an inclination of the regulatory expectations. The report should provide a level of detail which will enable a secondary evaluation by a regulatory assessor. And every population PK model will depend on the data and decisions made by the model developer, and every model has therefore unique properties. It's therefore vital that every assumption and decision made during model development is made clear for the assessor. And also that the analysis and report of the analysis needs to be of sufficient quality so that the final model can be judged to be a good description of the data, and that the results and conclusions ensuing from the population analysis can be considered valid. And I think that all of you obviously think this is obvious, but I assure you that all reports we see do not fulfill this. So the regulatory expectations regarding the analysis plan are described in this guideline. The analysis plan should be prospectively written and presented in an appendix of the report. And the components of the analysis plan are listed, and I will not go through these because you can read yourselves in the guideline. And when it comes to the report, this should obviously contain all the elements of the analysis plan, and any deviations from the analysis plan should be clearly stated. The introduction should put the analysis in perspective of other clinical data, and it's important to describe the results in detail, to describe the raw data, the covariates, the missing data and outliers, and describe the modeling and including a detailed description of covariate selection together with goodness of fit information and model qualification. And very important is that companies should critically assess the analysis. So the discussion should include a discussion on how well the final model describes the data, what the limitations of the analysis are, and the assumptions that, and all the assumptions should be discussed and justified. And the company should discuss what are the clinical relevance of the covariate influences, and are the covariate effects biologically plausible? How well do the results agree with previously obtained information? And how will the results in the analysis be used? These are another couple of quotes from the guideline. The amount and type of model qualification procedures will depend upon the objectives of the model. And model evaluation procedures to support an objective, that is to describe the data and evaluate potential covariate effects, could be simpler than those if the model is to be used for simulations, for instance, to support dosing recommendations. And the report should contain a justification for the model evaluation procedures. And if simulations have been done, these should be described in detail, including description of the demographics of the simulation data set. And we should remember that this guideline was written some years ago, and it was adopted in June 2007. So the guideline also has a quote that it's been written based on the current knowledge. And population PK is an evolving science, and this must be taken into account when interpreting the guideline. and it's expected that in the future also additional knowledge gained will be applied. So also, as Terry said earlier today, the level of assessment will depend on the importance of the analysis. So if you have a population PK analysis, which is the only source of evaluation of the effect of intrinsic factors, or if the analysis is used for dose adjustment in subpopulations, we will have a high level of scrutiny in the assessment. But if there are other data available to support the evaluation of intrinsic factors and potential dose adjustments in subgroups, or if there are no dose adjustments based on this analysis, the population PK analysis will be of lower importance, and we therefore will not assess it in such great detail. And reasons for failing the regulators' expectations can be deficiencies in the quality of the report. It may be insufficiently detailed. There may be missing information, or the report may be very detailed but may contain irrelevant information and lack important information. There may also be deficiencies in the quality of the analysis or in the underlying data. And the regulators may criticize the conclusions drawn from the analysis. Did I skip one? No. So my first example is an oncology product, a cytotoxic agent, where a body surface area-based dosing was applied in the clinical studies. And there were no specific studies evaluating the effect of intrinsic or extrinsic factors. And these were evaluated in an integrated evaluation of non-compartmental data and in a population PK analysis. And this analysis was dated in February 2009, and I just state that because then the guideline had been into operation for a couple of years, so it should have been known to the company. So the population PK analysis included 154 patients and had the objective to estimate PK parameters and inter-individual and residual variability in cancer patients, and to estimate the covariate effects. And the company used this analysis together with the integrated evaluation of non-compartmental data to evaluate the effects of intrinsic factors on the PK. And I will now come into some criticism by the assessor of this analysis. And this is very detailed sometimes, but it's just to give you a flavor of what kind of comments we may give. So the assessor criticized the exclusion of data because data below limit of quantification had been excluded, but it hadn't been stated how many data points had been excluded, and the assessor was concerned that if it was a large proportion of data, this would lead to biased parameter estimates. And also the company had excluded data which were unrealistically high, but they had not re-estimated the final model with these samples included. So the assessor was concerned that that might have affected, exclusion of these samples might have affected the model estimates. Further, the use of individual empirical based estimates was criticized. These were used in plots to illustrate covariate effects, etc. But shrinkage in this analysis was high. It was 29% or higher for different parameters. So the assessor considered that the conclusions made on the basis of the graphics were uncertain and inconclusive. And further, there was a concern that there was a model misspecification as high concentrations were not adequately captured. And regarding model development, weight was included in the model initially, but didn't result in a reduction of the objective function. So the assessor had a concern that either a local minima was found or otherwise body size was not an important predictor of systemic exposure. And this then would question the body weight or body surface area based dosing for this product. So the assessor concluded that the confidence for the model was low, and if the model were to be used in any responses to questions raised, then they would have to address all the issues raised and redevelop the model. So the regulator's concern here was that there was weak support for the data in the data for the relationship between clearance and body surface area. So there would be a risk for underdosing of low-weight patients and overdosing of high-weight patients with the proposed body surface area-based dosing. And the question was raised where the applicant was asked to discuss the risk for under and overdosing in these groups and how to ensure adequate dosing also in low and high BSA extreme patients with this low and high BSA. And I will not go explain what happened further in this application or in this procedure. It's still under revision, but this is just to illustrate an example of a deficiency in the quality of an analysis. The next example is a population PK analysis reported in May 2010, which included a large number of patients from 2 phase 2 studies. And also here the objectives were to define structural PK and PK/PD models and characterize inter- and intra-individual variability and covariate effects. And the company used these data together with other data to evaluate the effect of intrinsic factors on the pharmacokinetics. And the company conclusions were that covariate effects seem to be moderate and within the observed variability of plasma concentration time profiles of phase 3 patients, and that the effects were in line with those from separate dedicated clinical pharmacology studies. And they provided a simulation of a typical patient and with the 90% prediction interval around this in dotted lines. And then simulations of typical patients with varying covariates. So an elderly patient, a patient with low renal function, a patient with low weight, an elderly patient with low weight, and an elderly patient with low renal function. And they considered as all these concentration time profiles were within the 90% prediction interval for the population, that no dose adjustments would be needed. And I may have forgotten to say that the significant effects, covariate effects, were renal function and age and weight on volume, I think. Renal function and age on clearance and weight on volume. Well, the assessor criticized this evaluation because they didn't receive all the goodness of fit plots expected, and the high concentrations were not well described in the provided plots. Shrinkage was not provided, and the visual predictive check plots did not include all elements needed for proper assessment. And also here the conclusion was that if the model will be used for simulations in response to questions posed, the applicant would need to provide improved information on predictive properties. Regarding the simulations, these were criticized because they should include variability in the different subgroups, and the 90% prediction interval of the subgroup should be compared to the 90% prediction interval of the overall population. So the assessor did not consider that the evaluation was sufficient to claim that no dose adjustment would be needed in different subgroups. The company responded and provided additional information regarding the predictive performances of the analysis. Conditional weighted residual plots, shrinkage, prediction-corrected visual predictive checks, and all these were considered acceptable. So it was concluded that the model was considered to describe the data well. And the response also included a discussion on the effects of age and renal function with comparison to the results from dedicated clinical pharmacology studies. And based on this discussion, a dose adjustment was implemented in moderate and severe renal impairment. And this is an example of a deficiency in the quality of a report and criticism of the conclusions drawn. And all these could be resolved by questions and responses from the company and additional discussion on the need of dose adjustment. The third example is the population PK analysis reported in October 2010, which included data from both phase 1 studies with rich sampling and phase 3 studies with sparse sampling from a very large number of patients. And here the objectives were to characterize the systemic exposure in patients, identify covariates, and quantify sources of variability. And this analysis was used to evaluate effects of intrinsic factors on PK and inform the content of the SPC. And covariates identified in this analysis were creatinine clearance and gender on clearance and body weight on volume. And this assessor had concerns here because the covariate distribution was fairly limited. Regarding renal function, patients had mainly normal renal function or mild renal impairment. So the identified covariate relationship was considered unreliable and may not adequately characterize the true covariate, influence of renal function on this drug, taking into account more severe degree of renal impairment. And further, the age range was limited and did not include many elderly patients, so it was considered that we couldn't draw any conclusions in patients above the age of 70. Also, the visual predictive checks were difficult to interpret as they didn't include all the elements needed for proper assessment. And VPCs were stratified by dose, but not by subpopulations of interest, and there were no population predictive corrective visual predictive checks included, so we couldn't fully assess the VPCs either. And further, the use of empirical-based estimates was criticized because shrinkage had not been presented, so we didn't know if these were reliable or not. However, the effect of intrinsic factors was evaluated also in specific phase 1 studies and in a pooled analysis of phase 1 data. And the population PK analysis was therefore considered supportive, and there were no real discrepancies between these two evaluations. So therefore, no questions were raised regarding the population PK analysis despite the identified shortcomings. But the SPC was revised and any actual data from the population PK analysis was removed from the SPC. And this is an example of a deficiency in the quality of the report, the underlying data, and by that I mean the range of covariate distribution considering the conclusions the company draw, and also criticism regarding the conclusions drawn by the company because of the underlying data. The fourth example is a new modified release formulation, which was intended to be used in a indication for a replacement therapy where the current treatment is an immediate release product administered twice or thrice daily, and where the dose is titrated based on response. And the PK data available in patients here was from a clinical study with sequential crossover design. And the dose was individually titrated based on clinical response using the immediate release formulation. And this resulted in varying clinical doses between 20 and 40 milligrams daily. And after evaluating the PK for the immediate release formulation at these different dose levels, there was a switch to the corresponding daily dose for the modified release formulation, and thereafter a pharmacokinetic evaluation. And it turned out that the AUC was similar at the 25, 30, and 40 milligram dose levels. And this was applicable both for the immediate release formulation and for the modified release formulation. And these data were evaluated also by population PK analysis, which had an objective to describe the PK, to simulate other treatment regimens, and to develop dosing nomograms based on patient weight or concentration. And these data were used to support the dosing recommendations. The assessor had concerns here because the population PK analysis included the nonlinear exposure was modeled as nonlinear bioavailability. So the decrease in there was a decrease in bioavailability with increased dose. But the study design was a titration of the dose based on clinical response, resulting in similar AUC in the dose range 25 to 40 milligram. So patients with a higher oral clearance were likely titrated to higher dose levels, and this was not taken into account in the model. So the conclusions drawn were that the estimated dose bioavailability relationship was not reliable. Simulations of increased doses were unreliable, dosing nomograms based on this model were not accepted, and this is an example of deficiencies in underlying data. And additional data on for evaluating the dose proportionality in a crossover study would have been very helpful here to add additional information on this and solve these uncertainties. Examples of comments for other products are that often shrinkage is not reported even though the individual base estimates are used. VPCs often don't contain the elements we need to assess if the model is appropriate. And there may be limitations in the data. The range and distribution of covariates may be sufficient. This is usually the case for age, where very elderly are not included, or renal function, and race, which many studies include mainly Caucasian and very few subjects with other races. And also when it comes to concomitant medications, there is often insufficient information regarding dose level and duration of dosing for the concomitantly administered drugs, so it is very difficult to draw any conclusions regarding the influence of concomitantly administered drugs. My final comment is, though, that there are many population PK analyses that are of good quality, but that was not the topic of my speak. And there are several reasons for failing regulators' expectations, and these are often related to an insufficient quality of the report, and this is handled by questions raised. But a better quality of the analysis and reports would make the review process more efficient. Assessment of an analysis could then be made based on the report provided with the marketing authorization application. There would be no need for questions, and there would be less burden on both companies and regulatory authorities during the review process. So my recommendations are that reports should be sufficiently detailed, include relevant components, and model qualification is very important. And you must make sure that a secondary evaluation by regulatory assessors is possible with the data you provide in the report. And you should critically assess the analysis, discuss how well the final model describes the data and what the limitations of the analysis are, and if assumptions have been discussed and sufficiently justified, and what the clinical relevance of the covariate influences are, and how well the results agree with previously obtained information, and also how the results will be used. Thank you.
Thanks. So any questions of clarification for Monica? It's interesting that... yes, go on, Dr. Joseph, please.
I have one question, actually, and I hope I understood the example C correctly, that the assessor had some questions on the POP-PK analysis, but at the end, there was no specific regulatory question that was related to POP-PK analysis. Is that correct?
Yeah, I think I'll have to look which one was number C. Yeah. We identified a number of deficiencies, but we didn't raise any questions because we found sufficient other data to support the evaluation of intrinsic factors.
Okay, so actually it will be interesting for me also to hear from industry as well as from regulators, where in these cases where we probably, and in my experience at least with reviews, very upfront we know whether there's going to be a critical regulatory decision based on this model, and whether it is valuable for regulators to go into details about this particular analysis, and also is industry expecting us to review that? Because I was talking to somebody on exact these lines at the break, and at least my view in our division is I do not get into reviews of this application, but again, I would like to hear if there is a value on doing such reviews and critiquing the model, because it's resource.
So we'll take that forward into the discussion. Gerard, do you have a specific question?
Yeah, it has to do with validation of the models. I mean, having the experience of recommending modeling and simulation in the drug development in children at the PDCO, I'm always concerned about what is going to be done to validate our recommendation. And my concern is not so much the mathematical validation, but rather to check at the end what is the consistency between what is predicted and what is observed. And I think what I would call the clinical validation of the model. So could you comment on that?
Well, that's what we say, that model qualification is very important, and we need to have VPCs to really show that the model can describe the data very well. That's the very most important thing with these analyses, I think. Or did I misunderstand you?
Maybe not quite, but what when everything is done, we set up a model, we use it, we predict results, and what is going to check whether this prediction meet the observations. You know, it's not a mathematical intrinsic verification, it's just verifying at the end that the regulatory recommendation of the PDCO, for example, to be concrete, it has been valid.
I can see a number of reactions to your line of questions. So again, we're going to save it and we'll come back during the discussion for a wider view on that. Questions of clarification to Monica. Peter?
It's really just around the policies around refreshing and updating guidances. Is there a natural cycle time for refresh rates? The reason I ask is because a lot of the deficiencies that you were looking for seem to fall into fairly consistent elements that you were looking for: VPCs, CW-res, shrinkage, and they are scientific developments that have probably emerged after your 2007. Well, generally
some of them, but some shrinkage and CBRS are in the guideline, but they're not reported anyway, so people don't read the guideline. But VPCs and these percentiles of observed and the confidence interval and so on, that's not in the guideline. But guidelines are revisited, but not very frequently, and that there needs to be a sufficient number of elements that need to be updated for us to update the guideline. But maybe we need to do a small revision here.
There's a standard timeline that we would standardly look after 5 years, but we can look earlier and we can leave a guideline unchanged for longer if on reflection we think it still serves its purpose. So in theory you can open the guideline for revision at any time. The default to make sure we look at them is 5 years.